Okay, so in this video, or this chapter, we're going to be looking at what we call phylogeny and systematics. And so it's really looking at how these evolutionary relationships work and we're kind of make some diagrams to kind of demonstrate or graph the evolutionary relationships. So the study of phylogeny is looking at the evolutionary history of a species. Or group of related species. So you can look at all the mammals or all the vertebrates or something like that. Okay? And they use mostly the info that's used to construct phylogeny. It's two things, and some of those evidences of evolution that we looked at in chapter 15. It's the fossil record is the main one, or that's what they used to use, and all that anatomical structure and all that kind of stuff. And now the new thing is looking at molecular data. And so they look at DNA, RNA, proteins, and we're actually going to kind of briefly touch on that a little bit here, and then later in the year we'll understand that really in depth, and actually we'll look at your phylogeny a little bit, and we'll look at trace your evolutionary history a little bit in terms of humans using your own DNA, which is kind of a cool thing. So systematics is just the study of biological diversity in an evolutionary context. But we're going to actually, the next unit that we look at is all biological diversity. So it's, what's a proteus, what's a bacteria, what's a mammal, what's a fungus, all those kinds of things. But this is really looking at it in terms of an evolutionary context. And you guys know enough about it to kind of get there. And really, the goal of systematics is to trace phylogeny. Those are just definitions of words, guys. Don't obsess about these words, but just kind of understand. Now, what systematic biologists do is they use these tools called cladograms, and there's a couple cladograms here, and you can see the word cladogram. So a cladogram, all it is just a diagram, okay? It's just a diagram of evolutionary relationships. And we're going to learn how to make these and draw them and analyze them and all that kind of stuff, okay? And so you have branches or clads. So a clad is just a branch or a group on the cladogram. Okay, so if I were to circle in this cladogram over here, if I were to circle, this would be your common ancestor right here. Letter A is going to be the common ancestor of all of these different organisms up here, okay? These guys right here are part of a clad or a branch, and they all branched off of this organism B, okay? This clad actually also has something else going on for it. Besides just being a clad, this one that I circled here happens to be monophyletic. And what we mean by that term, is that it consists of ancestral species, and all the descendants. So you can see that it consists of the ancestral species and all of its descendants up here, okay? So that's a monophyletic group. Let's compare that to something that's not monophyletic. And then we'll look at a real-world example there, okay? So if I were to circle, I'm going to change colors just so you guys can really see this. If I were to circle just these top ones here, this would be not monophyletic, because I'm missing the ancestral species. I'm not circling these guys down here. I'm missing C and F and B, okay? So that's why it's not monophyletic. So let's look at an example. Here we have a lizard and a snake and an alligator or crocodile and a bird, okay? If you were to tell me, and if I were to ask you, what are these, what groups are these all in, you would say this is a reptile, this is a reptile, and this is a reptile, but that's a bird. Okay, so these guys are all part of Reptilia. Okay, or the class Reptilia, or just they're reptiles, that's fine. Okay, the class Reptilia is not monophyletic. Not monophyletic. Because the crocodiles and the alligators are actually more closely related to birds than they are to lizards and snakes. Okay, so that's kind of how they get that. So let's figure out how we actually make these diagrams. How do we get there? Okay, so we're going to learn how to construct a cladogram. So that's the next page. We're going to do constructing of a cladogram. And so really the first thing we want to do is figure out, we're going to try and sort homology or homologous structures, so from chapter 15, from analogy or analogous structures, okay? So remember, homologous have a similar ancestry. And structure. And the more homologous parts, more related. More homologous parts equals more related. They're more similar to each other, so they're going to be more related to each other, okay? Just as a jot, to jog your memory, analogous structures, if you remember, are different structures with the same function. And they really have nothing to do with each other in terms of evolutionary history. Okay, they just solve the same problem with a similar way, but they did not evolve together. So we want to get rid of those because they don't really tell us anything about a relationship. And in fact, these actually confuse scientists for a long period of time. But okay. The next thing we're going to do, the next kind of step is, and a lot of times we don't actually have to do these steps when we're constructing them in this class because they're already done for us, is we're going to identify shared characteristics or characters. Okay? So things that are specific to that group. We want to look for things that are specific to the group being studied. If, for example, we were looking at mammals and we said, oh, they have legs. Well, so do birds, and so do reptiles, and so do amphibians, and so there's a lot of things that have legs. So do insects. There's a lot of things that have legs. But what we want to look for, evolutionary novelty. Let's see if I can spell. Or things that are unique to that clade, okay? We're looking for things that are unique. And that's the hardest thing because you want to make sure that nothing else has it. So for example, jellyfish are in the phylum Cnidarian, we'll learn this later, but they have a stinging cell. Nothing else has a stinging cell. Or another example is hair in mammals. No other animals or organisms on planet Earth have hair. Okay, so that's unique to mammals. And then the last thing we want to do is we kind of want to compare it to everybody else. The outgroup comparison, we call that. Okay, so we're going to compare the outgroup. So an outgroup is species that are closely related, but not quite there, to the species being studied. Okay. And then what we're going to end up doing is we're going to do all these comparisons, and we're going to end up with this little matrix. We're going to create some kind of data matrix. We're going to determine our characteristics, we figured out who shares them, and then we've created this data matrix. So you can see here, there's things like hair and amniotic shells and walking legs and jaws and a backbone. Okay, and then you have animals across the top. And lancelet is actually going to be our outgroup in this example. They look kind of like a fish. They're actually chordate. So they have a backbone, but that's about it. They're not much else. We'll talk about them when we get to that section. But Then there's lampreys, which are jawless fish, tuna, a bony fish, salamander, an amphibian, a turtle, a reptile, and then a leopard, a mammal, okay? And you'll notice like a leopard has ones in everything here. That means it has all of those characteristics. If it was missing something, like a turtle does not have hair, so it has a 0 there, okay? And then what we can do is we can begin to construct our cladogram. So I'll show you. What you want to do is you just start with your common ancestor. You can even label it CA, common ancestor, write common ancestor. You don't have to label it. And then I like to just draw a long line like this. Sometimes they can get tricky though, so be careful. And the first thing to come off is our lancelet. It has nothing in common with anybody else, okay? So he's gonna go way off up here, lancelet. Okay? Then we're gonna have our lamprey come off. And this one happens to be in order, and a lot of times they are, but be careful, sometimes they're not in order. Then next is our tuna. And then there's the salamander. And I'll show you a trick here in a second. And then there's the turtle. Uh-oh. And then finally the leopard. Okay. Now, what you can see, oh, I can't see my leopard. Okay. Everything that's above this point right here has a backbone. Okay. A lamprey has a backbone, a tuna has all these things, they have backbone. And if you go over to your chart over here, you can see that all of these things have a backbone. Okay. And then everything above here has jaws. So a tuna, salamander, turtle, leopard all have jaws because you can see that on our chart here. Okay. So then right here, everything above that has 4 walking legs. And then everything above this has an egg, and then everything above this has hair. Okay? So let's try on the next page. Let's try another one, and then we'll talk about molecular clocks, but I might actually pause. This one is actually looking at some molecular data. And so they're showing you the number of differences in the amino acid sequence in a specific protein that's involved in cellular respiration in all these animals. More differences means less similar, okay? They're further apart evolutionary. So let's see if we can put these on here. So I'll actually start to do it, but I think that you guys might be able to figure out who has the most difference. So you might be able to answer that the snake has the most difference. So he's gonna go way out here. Okay. And then right here is the penguin, because he's got the second most difference. And then the chicken, and again, this one's in order. Not always are they gonna be in order. He's got the next most difference. And then the whale is gonna go here. And then the donkey. And then finally the horse. Okay. One question I always get is, can you switch the penguin and the chicken? Let's take a look. Let's look at the relationship between the chicken and the whale and the penguin and the whale. So the whale, compared to the chicken, has 9 differences, and compared to the penguin has 10 differences. So the chicken is a little bit closer. There's less differences, so it's a little bit closer to the whale than it is to the penguin. Okay? So you cannot change the chicken and the penguin. And the same goes with the donkey and the horse too, okay? Because of the differences between the donkey and the horse and the whale. There's 4, the donkey is a little closer to the whale than the horse is. Okay. The last thing on here is the idea of molecular clocks. Scientists can use these changes in phylogenetics and changes in evolution as a timing method.